So today we're going to talk about the male reproductive system, and by way of introduction I want to briefly talk about the development of testes, which is the main organ in the male reproductive system. The formation of testes in the embryo. So males have this is genetically determined. So males have an X chromosome and a Y chromosome. Okay, and females, which we'll be talking about in our next lecture, this is the symbol for females, have two X chromosomes. Something you probably already know. So, in the embryo, I'm talking about very early on in development, like in the 8th or 9th week of development, there is up in the abdominal pelvic cavity some tissue called the indifferent gonad. Indifferent means it has not differentiated yet into male or female, into testes, or the analogous organ in females is the ovary. Those are the main organs that make the gametes and hormones. But at first it's called the indifferent gonad. So gonad is a term that applies to testes or ovaries. So it's undifferentiated at first. So this same tissue is going to become either testes or ovaries. And it depends on basically one thing, and that is the Y chromosome. So the Y chromosome has genes on it that are needed for the development of the indifferent gonad into testes. The female doesn't have a Y chromosome and lacks those genes. They are not on the X chromosome. So in females, the testes are not going to develop. And in the absence of the Y chromosome, the indifferent gonads just sort of automatically differentiate into ovaries. So you could say in some ways that ovaries are the default gonad. In other words, that's what the indifferent gonad is going to become, unless there's a functioning Y chromosome. But for females, you don't need the X chromosome. In other words, there's no genes that need to be expressed in the female. You just need no Y chromosome to redirect the development in the testes direction. Okay, so we say that testes and ovaries are homologous structures because they develop embryologically from the same tissue. Okay, now once testes and ovaries develop, what about the rest of the male and female reproductive system? Because there are also sperm ducts in the male, there's glands like the prostate gland, there's the scrotum and the penis, whereas in females there's uterus and fallopian tube and other organs associated with the female reproductive system. So what is it that leads to differentiation of those in the male or female direction? And this is a bit of an oversimplification, but in general, that is guided largely by the production in the testes of testosterone. So in the young male embryo, there is a window during development when the testes makes testosterone. Okay, this is in the embryo before birth. It's not even a fetus yet. But testosterone is produced for several days or a week, and that leads to the development of the other, what I'll just call male genitalia, the sperm ducts and the glands and so on. And in ovaries, again, it's the female is more of the default sex. In the absence of testosterone, you get female genitalia. I'm talking about ducts and uterus and glands. So that's a little overview of the embryology of male versus female reproductive systems. Okay, so today though we're just going to focus on the male, and we'll talk about the female in the next lecture. So here are the organs that make up the male reproductive system. It's divided into two types, internal and external genitalia. Internal just means you can't see them. They're not superficial. They're within the body. External, you can see them externally, superficially. So external genitalia would include the scrotum, which houses the testes, and the penis. The internal genitalia includes the testes, which is the main organ, as we were just talking about. The testes is where you produce sperm and hormones like testosterone. So they call it the primary gonad. The primary gonad, the main organ of the male reproductive system. And then here are the four ducts: epididymis, ductus deferens, also called vas deferens, you can use either term, the ejaculatory duct, and the urethra. Those are the four ducts that carry sperm. And then we have three exocrine glands we're going to talk about: seminal vesicle, prostate gland, and the bulbourethral gland, sometimes called Cowper's, C-O-W-P-E-R, Cowper's gland. So mainly we're going to be focusing on the function, although we'll look a little bit at the histology. So here's a picture of all these organs. The testes, as I was mentioning briefly previously, the testes develop from some indifferent tissue up in the abdominal pelvic cavity. But before birth, they need to descend not only into the lower pelvic cavity, but out of the abdominal pelvic cavity into the scrotum. So the scrotum is a muscular sac that houses the testes inside. So it's lying out of the pelvic cavity. The reason for this is that for whatever reason, sperm production, which happens in the testes in males, requires a lower body temperature than up in the pelvic cavity. So it's for lowering the temperature. And the scrotum, which has muscle in it, helps regulate that. If the testes get too cool, the muscles will contract and pull it closer to the body to raise the temperature, and then vice versa. If it's temperature's too high in the testes for sperm production, they'll relax. So the scrotum is not just protecting the testes. It has a regulation of temperature as a function. Okay, so that's the testes, and then let's follow the ducts, the four ducts in order. The epididymis is the first duct. It's right next to the testes, and it also stores sperm that are made in the testes, so the sperm can be stored there. And then during what's called ejaculation, the smooth muscle contractions in the ducts are going to move some, not all, but some of the sperm out of the epididymis, and then up a very long duct that comes all the way up here, loops around the bladder, comes around, and stops right here. This is sorry, it's hard to see my red pen. This is called the ductus or vas deferens. Okay, this is the one they cut in a vasectomy. So that's the second duct. And then if you look closely at the very end of it, where it's a little bit wider, they call it the ampulla of the ductus deferens. Notice how then it merges with another duct coming from this gland right here, the seminal vesicle. So you see here how these two ducts are coming together. Once they merge, we get into the third duct. They call it the ejaculatory duct. It's called that now because the sperm that was in the ductus deferens is going to be mixed with secretions from the seminal vesicle and also another important male exocrine gland, the prostate gland, which is right under the bladder. And it makes various products that, in addition to the seminal vesicle, get secreted into the ejaculatory duct, and with mixing with the sperm to form semen, S-E-M-E-N. So semen is sperm plus the secretions from these glands, prostate, seminal vesicle. So that's why they call it the ejaculatory duct, because that is the material now that's going to be released shortly from the male, a process called ejaculation. Okay, so the ejaculatory duct comes through. It's also not very long, like the epididymis, and then it meets the fourth and final duct, which is quite long, and that is the urethra. So we looked at this since we've already covered the urinary system. So let's just go back to the bladder for a minute. So here's the bladder up here. So urine leaves the bladder through the urethra. But notice that the ejaculatory duct also enters the urethra right in this area. Now, urine and sperm are not going to go through at the same time. This is all controlled by sphincters that can close off. For example, there's a urethral sphincter near where the bladder empties into it that you can close off when sperm are coming through to keep urine out of the urethra. So they're not traveling through at the same time. Okay, so sperm's going to enter the urethra a little bit down from where it begins, and then the urethra is going to go through, well, a little part of it's going to go through the prostate gland, not much of it, but they call that the prostatic urethra. And then it's going to, another short little section is going to go through the muscular floor of the pelvic cavity. They call that the membranous urethra. So that's the second part of the urethra. And then as soon as it enters the penis, there's quite a long part of it coming through there. That's called the spongy or penile urethra. So it's just one duct, but it's so long they divide up into those three segments: prostatic, membranous, and penile or spongy urethra. Okay, so once again there are four ducts: epididymis, ductus deferens, ejaculatory duct, and the urethra. And then you can talk about the three parts of the urethra. Okay, so those are the ducts, and then we looked at the two glands. And then there also is another gland you need to know, the bulbourethral gland. Here it is. It secretes into the urethra, but it's not forming semen. It's mostly secreting an mucus that's sort of coating the urethra before the sperm come along. Okay, so let's start with the testes. Two main functions: sperm production and making testosterone and other androgens. So this is the primary gonad. Here's an actual testes, and you also see the epididymis right here. And then the epididymis, if you remember, leads into the ductus deferens, but it's hard, you can't actually see the ductus deferens here, but it is coming up this way, and it's traveling with blood vessels, lymph vessels, and nerves, and they're all wrapped with some connective tissue. So it's in there, but you can't really see it. They call the whole thing the spermatic cord. Okay, so here you can see it better. There's the ductus deferens coming up through here, along with blood vessels, etc. And then let's look at the testes. It's sort of like an orange with sections. It's sectioned with connective tissue septa that's separated into compartments called lobules. And here's the main thing you need to know. The main structure in testes are called seminiferous tubules. We actually looked at these back when we looked at the testes for the endocrine system. So you might remember this slide that's going to be coming up soon. So the seminiferous tubules are where the sperm are produced. Okay, so they're the main structure in there. And then in between the seminiferous tubules are the cells that make testosterone. So here's the slide that hopefully looks familiar to you. This is a cross section here of a seminiferous tubule. So this whole thing here is one seminiferous tubule, and it has a lumen. And then you see all these dots here in the wall. Those are the cells that are forming sperm. The sperm forming cells. On the periphery of the seminiferous tubule, out here on the edge, is where the stem cells are. They're called spermatogonia. the stem cells, the ones and the only ones in the male that can undergo meiosis to form sperm. Now remember, meiosis is all about halving the number of chromosomes. So for humans, going from 46 to 23, so that those can form the gametes, the egg, and sperm in males, eggs in females. And then if they fuse, then you restore the 46 chromosome number characteristic of humans. So spermatogonia, if you remember from general biology, 2N means having a full set of homologous chromosomes, or 46 chromosomes in humans. Okay, so anyway, that's where the stem cells are located, on the periphery. They're not in the lumen, they're not in the middle, they're out on the periphery. And then as they undergo meiosis to form sperm, the various stages of meiosis, prophase, metaphase, anaphase 1, etc. As the cells go through that, they get closer and closer to the lumen. And then by the time they're here in the lumen, they are finished with meiosis, and they are haploid, meaning they have only half the number of chromosomes. Haploid cells called spermatids. So that's mainly what's going on in the seminiferous tubules, are the production of sperm. Now it's pretty easy to see the sperm in this picture because they're undergoing meiosis. Their chromosomes are condensed, and you can see them. They stain quite darkly. What's hard to see, ironically, are much larger cells that are in the seminiferous tubules called Sertoli or sustentacular cells. So I'm going to show you a picture where you can see this better. So in this picture, just to orient you, the lumen is down here. And this is the wall here of the seminiferous tubule. So again, you see all these little dots in there are the various cells undergoing meiosis. And notice they have names at every stage. Okay, the spermatogonium are the stem cells, and then after they go through meiosis 1, they're called secondary spermatocytes, and then when they're done with meiosis 2, they're called spermatids. So anyway, like I was saying in the previous slide, as they go through meiosis, they go further, closer and closer to the lumen. Okay, but now here are the other cells. The other cells are the Sertoli cells, and they are quite large, and they extend all the way from the edge of the seminiferous tubule to the lumen. So this orange color you see in here, this is the cytoplasm of the Sertoli cells, and here's a nucleus of one of them. So we only see a few Sertoli cells in this picture. As they extend again all the way from the periphery to the lumen, so they're quite large, and you see these white spaces around the developing sperm. Those would be the space in between the developing sperm and the Sertoli or sustentacular cells. And these cells are very important, these Sertoli cells, because they protect the sperm. They protect the sperm from antibodies in the blood. So in between the seminiferous tubules, like I'm going to draw a couple, here's some seminiferous tubules. There's 4 of them. In between are the, well, we'll talk about this a little later, but that's where the cells are that make testosterone, the Leydig cells. So we'll review that in a moment. But also there's blood vessels in here. And the antibodies in those blood vessels would, if they could, attack the developing sperm. And so the Sertoli cells protect them from that and form what's called the blood-testis barrier. So for something to get from the blood to the developing sperm, you have to go through a Sertoli cell. So they're guarding the developing sperm. And they're doing more than that. They're also providing some nutrients. They have regulatory roles. Actually, here's all their many functions. So forming the blood-testis barrier, nourishing the sperm. They can phagocytize, so they can protect in that way. They help regulate the movement of the sperm. They secrete a hormone called inhibin, which we're not going to be covering, but it affects FSH levels. So, they have many important roles. So there's their alternative name. Sometimes they even call them nurse cells. They're like nursing the sperm. Okay, so spermatogenic cells just refer to all the cells forming sperm, the stem cell and all the intermediates. And then here are remember these from the endocrine system, the Leydig or interstitial cells between the seminiferous tubules that make testosterone. So once again, that's the testes. It's making sperm in the seminiferous tubules, and it's making hormones in the cells between the seminiferous tubules. So here we see the same thing again in a different picture. Here are the stem cells. Every time you want to use a stem cell, it first has to go through mitosis so that you keep a stem cell, so that you don't use up your stem cells. So, one of the daughter cells will remain as a stem cell, and then the other one will undergo meiosis to form the haploid spermatids. Okay, so that process of meiosis, the fancy name for it in males is spermatogenesis. So spermatogenesis is meiosis in the male. Genesis means creation of spermato, means sperm. Creation of sperm through meiosis. Okay, so meiosis or spermatogenesis occurs once again in the seminiferous tubule. Now, if you look at a fully mature sperm, as you see down here in the lumen, they do not look like the spermatids, do they? Spermatids are round, haploid cells with 23 chromosomes. So really, once you have spermatids formed, meiosis is over. But the cell is going to transform from a spermatid to these elongated cells called spermatozoans. And that happens after meiosis. It's called spermiogenesis, the formation of sperm. Well, sounds like spermatogenesis, doesn't it? It's confusing. Spermiogenesis has nothing to do with chromosomes. The meiosis is done. The cells are already haploid. Spermiogenesis is more about morphological changes in the cell. It's going to change shape. It's going to grow a flagellum that it's going to need if it's introduced into the female reproductive system so that it can move along, and a few other features we'll look at in a moment. So going from a round spermatid to an elongated spermatozoan occurs mainly in the lumen of the seminiferous tubules and also in the epididymis. It can finish up in the epididymis. That's spermiogenesis. So let's look at sperm. We'll look at a picture in a moment, but here's some facts about it. So in the life of a healthy male, which starting at puberty means when does the reproductive system become functional. So somewhere in early teens usually. That's when sperm start to be produced. And then in males, sperm can be produced for as long as the male is healthy. It does tend to decline with age, but unlike females, where there's a point where the ovaries stop producing eggs, menopause, the same is not true in males. So, between puberty and as long as the male is healthy, about 300 million per day are produced in a kind of youngish middle-aged male, and then it will decline. Once a sperm is formed and introduced into the female, it can only survive a couple days in the female reproductive tract before it dies. So it has about 2 days to fertilize an egg. We'll talk more about that after we cover the female reproductive tract. So we'll come back to that later. And then here's what we're going to talk about next, the parts of the sperm. So usually sperm are divided into a head. Let's look at the picture. the head of the sperm, and then some books talk about the mid-piece or the neck. In this picture, they're just including that with the head, so head and neck, and then the tail, the flagellum. Now recall, flagella are elongations of the cell. They contain various proteins in them, like microtubules, that allow them to whip back and forth and allow the sperm to swim along in a fluid environment. Now, I want to stress that sperm do not use their flagella while they're in the male reproductive system. When they move through the ducts, they are not swimming through the ducts. They are being moved along just like you move food through your digestive tract or urine through your urinary tract. They're moved by peristalsis, rhythmic, smooth muscle contractions of the ducts. We're not going to be going into the histology of the ducts in lecture. In lab, I think there's a slide of one of the ducts. We can see there's an epithelial lining of the lumen, there's some connect tissue under that, some smooth muscle. So it's a typical organ in that respect. So sperm do not need their flagella until they're introduced into the female reproductive system, and then they have to swim up through the vagina, through the uterus, to where the egg is, which if it's there at all, it would be in usually the fallopian tube. And again, that's material we cover later. So the point I'm trying to make right now is just that the flagella is necessary in case the sperm ends up in a female reproductive tract. Oh, forgot to mention one thing about this. So let's go back to the head. The sperm head has two main important things in it. Of course, the nucleus, which you could say is the most important thing, is this has the half of the chromosomes that ideally, for the sperm standpoint, would unite with 23 other chromosomes from an egg cell to form a new offspring or zygote. So the chromosomes are in the nucleus of the sperm. And then you see this structure called the acrosome. It looks like a boomerang over the head. That is a sac of enzymes, acrosomal enzymes, that this sperm will need if it encounters the egg. Because the egg is not going to be alone. It's going to be surrounded by some other cells, and this sperm would need to get through that barrier to get to the egg. So it's going to need those enzymes in there. So again, that's a topic that we're going to cover in a later lecture, the release of the enzymes from the sperm head. So we do talk a little bit in this class about fertilization. Okay, now let's look at the glands. Seminal vesicle, prostate, and I've already said everything I'm going to about the bulbourethral or Cowper's gland. It's secreting mostly mucus into the urethra ahead of the sperm to kind of coat it, and maybe also neutralize the pH a bit because urine tends to be somewhat acidic. Okay, but these other two are contributing to semen. They're going to mix with the sperm to form semen. So here are some things that are in the seminal vesicle. We're not going to talk about each and every one, but I did want to mention a few. Fructose appears to be the nutrient of choice for sperm instead of glucose. So fructose for energy, right, for making ATP. There's an alkaline fluid. You want to dilute the sperm, and the pH is a little bit high in here because when the sperm are introduced into the female reproductive system, if they are, there the environment is on the acidic side. So having an alkaline fluid will kind of help neutralize the pH for the sperm. Prostaglandins are hormones that their role is not entirely understood, but it's thought that these hormones, which these are not any hormones we talked about when we study the endocrine system, but these hormones are thought to help the sperm travel in the female reproductive system from the uterus up to the fallopian tubes. Okay, and then there's for the prostate gland, there's I wanted to mention this protease, PSA, prostate specific antigen protein. Okay, this is a normal component of semen, and what it does is it makes the semen more liquid. It helps make it more fluid, and that helps the sperm become more mobile, more modal. And they also think that these PSA proteins help the sperm, if again they're introduced into this female reproductive tract, helps them get up through the cervix, where there's often a kind of a mucus-like plug or that the sperm have to get past. And then it's got some other enzymes. Pepsinogen breaks down proteins into peptides, some other enzymes. So those are some of the main components of these two glands that are going to mix with the sperm. Oh, and here's the Cowper's gland. So notice it's mostly mucus. Okay, so in general, semen, here are some statistics about it. Per ejaculation, which means how much sperm is released through the urethra per ejaculation, is about 2.5 to 5 milliliters. And then in each milliliter, this is about how many sperm there are. So just to give you an idea of, the range of how many sperm, there might be 250 to 600 million or so sperm in an ejaculate. Okay, so before I talk about this, sorry I'm doing this. I want to go back to this picture way back here. I did want to mention something about the penis. So I've talked about the scrotum's function in thermoregulation. What about the penis? The penis has, and this is something you'll look at in lab, it has a special kind of tissue in it called erectile tissue. Erectile tissue. Erectile tissue, you can see it in blue. Well, it's hard to see, but it's this part called the corpus spongiosum around the urethra, and also this corpora cavernosum. All of that is erectile tissue. Erectile tissue has spaces within it that when the penis is not erect, the spaces are empty, but during the erection of a penis, they fill up with blood. This is controlled by the autonomic nervous system. So those spaces in here, these little cavities, fill up with blood during an erection. That makes the penis erect. And the purpose of that is that the penis is what they call copulatory organ in males. Its job is to introduce the sperm into the female reproductive tract. So in order to do that, it has to transition from a flaccid to an erect condition, and then it can be inserted and ejaculate the sperm into the female. So that's the function of the penis, to introduce sperm. So back to my last slide, or next to the last slide. This is something you already learned, so this should be a review. But it's going to come up again on your final, so review it if you need to. Remember that the main hormones that regulate testes and ovaries are FSH and LH, made in the anterior pituitary gland. Remember that. If you have, here's the hypothalamus up here. If you recall, that makes a hormone called GnRH, although you might have learned it as FSH, LH releasing hormone. But now I would like you to learn its correct name. GnRH, gonadotrophin releasing hormone. It's on the next slide if you want the spelling. Anyway, that's made in the hypothalamus, and then it goes down to the pituitary and stimulates FSH and LH both. Recall that these two hormones share a releasing hormone. And what do those hormones do? Well, to remind you, you'll also have to know it for females, but I'll review it when we get the female when we get to that. But let's just focus on males right now. So in males, FSH is needed for sperm production. All right, that's a little vague, but it's needed for meiosis in the sperm. And LH is needed to trigger testosterone production. So the more LH that's secreted, the more testosterone is made. And testosterone is necessary in the male. It's a necessary hormone. I mentioned it way back in the embryo, where it is needed for a very short period of time, but it's absolutely necessary for the development of the male internal and external genitalia. Not the testes, but the other structures. But after that early window of release, the testes shut down, and testosterone will not be secreted again for many years, not until puberty. So, and then starting at puberty, testosterone production is one of the changes you see happening at the onset of puberty. The testes start making testosterone, and that testosterone has many effects. First of all, it brings about what are called secondary sex characteristics. Secondary sex characteristics. These are not organs. These are traits, characteristics that we associate with maleness. Increased muscle build-up in the body, skeletal muscle, facial hair, thickening of vocal cords, axillary and pubic hair development, behavioral changes in the brain. These are all referred to as secondary sex characteristics. And but beyond that, testosterone is absolutely necessary for the not just the development of the male genitalia, but also the maintenance of them. So males need to keep making testosterone in order for sperm production to occur and to maintain the sperm ducts and glands. All right, so that completes our lecture on the male reproductive system, and in the next one we will cover the female, which is more complicated because in addition to making egg cells and hormones, the female has to also provide a site for the development of the egg in case it gets fertilized. So that makes it more complicated.